Imagine diving into the deep ocean, exploring a world that's largely unknown. Beneath the surface lies an immense diversity of life, much of it yet to be discovered. Scientists can collect environmental DNA or eDNA from these waters, which contains the genetic fingerprints of the organisms living there. However, analyzing this eDNA is a huge challenge. The deep sea ecosystem is complex, species are largely undocumented, and traditional tools are not built to handle the scale and novelty. Our project, Blueprint, aims to unlock the secrets hidden in the deep sea eDNA by providing a fast, accurate, and scalable analysis platform that can detect even species that have never been recorded before.
Now, let us walk you through Blueprint. So, initially, after extracting the eDNA sequence from our underwater submersible, we will upload the .fasta file to Blueprint. Now, we can choose between the dynamic programming based algorithms like BLAST or Newman.
Current methods and limitations. Currently, scientists rely on BLAST, FASTA, and a few ANN algorithms like HNSW, FAISS, or ANNOY for DNA sequence similarity searches. While these methods work well for known species or small data sets, they struggle with two major issues. First, scalability. Analyzing billions of sequences is extremely slow. Second, novel taxa detection. These tools often miss species that are not in existing databases. In short, current methods are either too slow, inaccurate, or incomplete, essentially for deep sea research.
Thus, we can directly choose ZHNSW for the sequence matching instead and we'll wait about 3 to 7 seconds.
Why approximate nearest neighbor? When analyzing billions of sequence, traditional exhaustive search is too slow. Approximate nearest neighbor (ANN search) solve this by quickly finding the similar sequence to a query without even comparing with every entry. ANN drastically reduces the computation time while maintaining high accuracy, making this ideal for high-dimensional eDNA embeddings. So, building a ANN, we developed a ZHNSW. It's a zonal hierarchical navigable small word. As the dataset grow, traditional ANN methods slow down. So we use ZHSW, which uses zonal partitioning, splitting data into a smaller graphs for parallel search. And adaptive zone selection focuses on most relevant region, improving speed and recall. In benchmark, ZHSW is 3 times faster and achieves 98% recall and reduces 29% of their index size when compared to other ANN methods.
After this, we get a taxonometry of the genus, species, placenta, etc. of the respective eDNA sequence.
To achieve precise taxonomical classification, we collected around 2 TB of eukaryotic sequence data from PubMed and other public repositories. We processed this massive data set by first cleaning and standardizing the sequences, removing duplicates and low quality entries. Next, we extracted meaningful embeddings from each sequence to capture its biological features. These embeddings were then organized and indexed using a ZHNSW framework, which partitions the data into multiple zones for efficient similarity search. This indexing pipeline allows our AI models to quickly and accurately compare new eDNA sequences against billions of references, enabling high confidence species identification and discovery of novel taxa in deep sea samples.
As an additional feature, we have provided a 3D visualizer for research scientists to visualize the respective 3D sequences.
Why Blueprint is better? Blueprint is specially designed for deep sea eDNA, where reference database are sparse and many species are unknown. Our AI-powered classification uses multiple markers simultaneously, giving higher accuracy than single marker tools like BLAST or FASTA. The JET and SW search engine scales to billion case dataset, maintaining high recall bit rate while traditional ANN methods slow down. We are also integrate environmental and expedition data like depth, temperature, and salinity, enabling habitat-specific corrections that other platforms ignore. Finally, Blueprint provides real-time visualization, abundance estimation, and automated reporting. turning raw eDNA sequence into actionable insight in a single seamless workflow.
In short, Blueprint is faster, smarter, and more comprehensive than any existing solution for deep sea biodiversity search. Thank you for staying tuned to our video.